So what we did was we took a finding of MS patients and studied it in preclinical models of MS. And the clinical finding we looked at was that in metabolomics, a multi-center metabolomic study involving approximately 250 healthy controls and 500 MS patients, we found that indole metabolites are dysregulated. This has also been confirmed by other groups. And one of the top dysregulated metabolites was indole lactate, so short ILA. And then we took this metabolite and studied it in these preclinical models. We supplemented it and found that it dampens neuroinflammation and promotes remyelination. And I think this is the data I shared last time. Now, the update is that in the patients with MS, we see that not only does ILA, so indole lactate, change, but also another metabolite of this indole group changes, and that is indole acetate. In contrast to indole lactate, which is produced from tryptophan through a reductive pathway, indole acetate is produced through an oxidative pathway. And we saw that in patients with MS, ILA is reduced and these oxidative pathways are increased. So, and then the ratio of ILA to IAA correlated with disease severity. So now we, in our current studies, wanted to know what relevance does this change of the ratio of the two metabolites have. So for this, we again supplemented mice with ILA and found that, as expected, circulating levels of ILA increase, but we also see a decrease of circulating indole acetate. And then we asked ourselves, what is the relevance of this? For this, we investigated IAA, so indole acetate's effect on remyelination, and found that in contrast to indole lactate, indole acetate blocks remyelination. So to sum this up, our new finding is that ILA supplementation does not only block neuroinflammation and enhance remyelination, but also restores this imbalance of these circulating indole metabolites. Our next step, or what we're particularly interested in now, is investigating why indole lactate also alters circulating indole acetate levels. And both indole lactate and indole acetate are produced by the gut microbiome. And so we hypothesize that ILA might impact the composition or the function of different or the functional activity of different gut microbiome. And so yes, we're carrying out microbiome studies on mice treated with ILA.